This is your Cellular Respiration Notes, Chapter 8. Cellular respiration is how cellular organisms obtain energy by breaking down organic molecules, like carbohydrates, lipids, and proteins. Cellular respiration's goal is to harvest the electrons from carbon compounds to make ATP energy for the body to use, meaning we eat to fuel our bodies full of energy for all of our daily activities or demands that we put on our body. Here's the cellular respiration equation. You do need to memorize it, but again, you only need to know the words. Glucose plus oxygen are reactants. Mixed together will produce carbon dioxide, water, and ATP energy. So how does this compare to photosynthesis? Well, they're kind of flip-flopped. Glucose and oxygen were our products in photosynthesis. Here, they're our reactants. Carbon dioxide, water, and energy are our products here in cellular respiration, but were our reactants in photosynthesis. However, the energies were different. Light energy used in photosynthesis, ATP energy produced here. There are two types of respiration. One is aerobic respiration. This is where oxygen must be present. So if you think of yourself or me there doing aerobics, you need to breathe in oxygen in order to do it. Anaerobic respiration is on the flip, which is where oxygen does not need to be present, and usually it's not present. On the front of your note packet, you should see a flow chart of how cellular respiration functions. This should be memorized to aid in your understanding of cellular respiration. All boxes represent a process, and a cloud or star represents a product. So, glycolysis would be a process, right, since it's boxed. Pyruvate or pyruvic acid would be a product since it is starred. There are two main parts to cellular respiration when oxygen is present. However, the first step always, no matter what, is glycolysis. Then, once we get into aerobic respiration, our two steps are Krebs cycle and the electron transport chain. If oxygen is not present, then we would go through lactic acid fermentation. The first step is always glycolysis. This is when glucose is broken down in the cytoplasm. It is an anaerobic process, which means that oxygen does not need to be present. It's also enzyme assisted to help it make and go faster. Since it is a process, it needs some energy to get started, so we need to have 2 ATP to get us started. At that point in time, glucose is broken down into what we call a 3-carbon compound with phosphates attached at the sides. This carbon compound is called pyruvate or pyruvic acid, and we will produce 4 ATP. So our end result, though, is a net gain of 2, since if you do the math, we had to start with 2, so that's a negative 2. We produced 4, so that's a positive 4. That gets us a net gain of 2 plus 2 pyruvates and some NADHs.
Glycolysis, which begins the breakdown of glucose, is a series of 10 enzyme-catalyzed chemical reactions that can be divided into 2 main phases. In the energy investment phase, some ATP energy is used to start the process of glucose oxidation. By the end of this phase, a 6-carbon molecule, glucose, has been split into 2 3-carbon molecules of glyceraldehyde phosphate. The 3-carbon glyceraldehyde phosphate molecules now enter the energy payoff phase. Chemical bonds are broken, and NAD+ picks up electrons and hydrogen ions, forming NADH. The energy released is used to attach phosphate groups. The phosphates are transferred to ADP, finally making some ATP. This way of making ATP is called substrate-level phosphorylation. A couple more reactions rearrange the atoms in the 3-carbon molecules. More ATP is generated in the final reaction that yields pyruvate. For each glucose molecule broken down during glycolysis, A net of 2 ATPs are formed, along with 2 NADH molecules.
Hopefully that helped you understand glycolysis a little bit better. If you look at the diagram, we are in the cytoplasm. We're in the process of glycolysis. It is boxed. Our glucose is going to be broken down into 2 pyruvates. We needed 2 ATPs to get us started. We end with a net gain of 2 ATPs and some NADHs. We're next moving on to step 2 and to step 3. The Krebs cycle is when oxygen is present and we are now in the mitochondria of eukaryotes. In this stage, the pyruvate is going to be converted into a 2-carbon compound, which we call acetyl-CoA, and then it goes on through a series of more steps, which you don't really have to know, but I just want you to know that it does go through the cycle 2 times, since there were 2 pyruvates, and our end result would be 8 NADHs, 2 FADH2s, 2 ATPs, and 6 carbon dioxides. Again, remember that it goes through the cycle twice. This is the cycle, so our pyruvic acid becomes acetyl-CoA. It goes through the whole entire cycle and breaks it down for the next step of electron transport chain. And here is an animation.
The oxidation of glucose continues in the citric acid cycle. Pyruvate molecules formed during glycolysis diffuse from the cytosol into the mitochondrion, but pyruvate itself does not enter the citric acid cycle. A reaction occurs that removes a carbon atom, releasing it in carbon dioxide. Electrons are transferred to an NADH molecule, storing energy. Coenzyme A, or CoA, joins with the 2-carbon fragment, forming acetyl-CoA. 1 molecule of acetyl-CoA enters the citric acid cycle. The 2-carbon fragment of acetyl-CoA attaches to the 4-carbon molecule, oxaloacetate, in the first reaction of the cycle. This forms citrate. In a series of steps, bonds break and reform. 2 carbon atoms are released one at a time in molecules of carbon dioxide. Electrons are carried off by molecules of NADH and FADH2. One step produces an ATP molecule by substrate-level phosphorylation. A 4-carbon oxaloacetate molecule is regenerated. Since 2 acetyl-CoA molecules are produced for each glucose molecule broken down, a second acetyl-CoA enters the citric acid cycle. The same series of reactions occurs, releasing carbon dioxide and producing more NADH, FADH2, and ATP. The cell has gained 2 ATPs that can be used directly. However, most of the energy originally contained in the bonds of glucose is now carried by the NADH and FADH2 molecules.
Hopefully this helps you understand the Krebs cycle. Next, we have the electron transport chain. This is the last step of aerobic respiration. This is where most of the ATP is produced. Our NADH and FADH2s release their hydrogen ions to help create that ATP energy. Those hydrogen ions will move along the mitochondrial membrane. They're going to be pumped back and forth along that membrane, transferring the FADH2 into an FAD, releasing 2 hydrogens, NADH into NAD+, releasing their hydrogen. This causes more hydrogen to be released and to be pumped along the membrane. Something very similar to what happened in photosynthesis. This will end up creating 32 ATP. Some of the hydrogen ions unite with oxygen to create water, which is released by the body, and so our end result is 32 ATP with some water. So you can see our pumps, and then the hydrogen going in and out to ultimately create all of our ATP.
Cycle produces small give up their
is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward highly electronegative oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and two hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. This mode of ATP production is called oxidative phosphorylation because it is powered by the transfer of electrons to oxygen. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by electron transport and oxidative phosphorylation, about 34 ATPs for every glucose consumed.
It does say 34.
Cellular respiration. Glycolysis in the citric acid cycle.
Stop.
The last complex.
No, stop. Sorry, it does say 34 because it takes into account the 4 that were produced during glycolysis. It doesn't subtract them. But please make sure you know that in electron transport chain, it is 32, not 34. So here we have our cellular respiration diagram. Glucose is broken down the process of glycolysis into pyruvate. It then goes on to the Krebs cycle, and then onto the electron transport chain, where our ultimate energy maker is. So the end products of aerobic respiration. Glycolysis produces 2, Krebs cycle produces 2, electron transport chain produces 32. So from one single glucose molecule, we're getting 36 ATP. So why in the world does this make sense? Well, obviously our bodies need energy to do anything and everything. Breathing, sleeping, digestion, thinking, blinking, you name it, and energy is needed. So getting 36 ATPs per glucose will give that organism plenty of energy to do whatever it needs to do. And in actuality, if you look here as a trivia fact, the body uses about 1 million ATP molecules per second. Prokaryote aerobic cellular respiration. Well, they don't have a mitochondria, so they actually just use their cell membrane and use that as a pump, and they end up making 38 ATPs. And that ends your aerobic respiration notes.